Hi, everybody, and welcome back. Let's dive into Chapter 8 Cellular Reproduction, or How We Get Cells from Cells. Cell theory is an overarching tenet that all scientists adhere to. There really isn't much debate about the components of the cell theory, and this was postulated in the 1800s by two scientists, a doctor and a plant scientist or botanist, Matthias Schleiden and Theodor Schwann, as they were doing their research and studying cells and cellular structure. They came to the conclusion that all living things are made of cells. Cell is the basic unit of life, and all cells come from pre-existing cells. So make sure you know all three components to the cell theory, and one of those components can potentially be problematic in terms of worldview. So number 3 states that all cells come from pre-existing cells. That may lead to the question of, well, where did the first cell come from? We know from that first cell, we can say with confidence all other cells have come from that. But the question of whether that first cell was either created or evolved, that is dependent on your worldview. So from there, science is able to answer how other cells have come to be, but it doesn't answer how the first cell came about. There are only theories, of course, so just keep that in mind. In terms of cell division, so what's the idea? So it's just how a cell reproduces, a kind after its own kind. Two main things happen. Everything in the cell has to double, including our genetic information in the form of chromosomes. And then each new cell is then called a daughter cell. And if we're talking about mitosis, the daughter cells are identical to the parent. So mitosis is one of the two types of cell division that we are going to be exploring here in Chapter 8. It serves to replace old or damaged cells. So, for example, if you get a scratch on your arm, that scratch will heal itself because your skin cells surrounding that damage or cell loss, essentially, will grow to replace that lost area of cells. Growth as well. So think about yourself as your zygote form, or your one-celled self. We all started off as one cell very early on in our life, but from there we underwent mitosis many times to grow into fetuses, babies, children, and the adults that we are now. So mitosis is asexual reproduction, and so some organisms are able to clone themselves, essentially. So that's what that means. Asexual, no sex involved, requiring only one individual instead of two. And again, mitosis is basically copying and pasting cells. There is nothing different, or there's nothing supposed to be different at the end versus at the beginning. So we're just trying to maintain growth, development, and cell replacement here. Sexual reproduction, however, requires two individuals. So most living things do sexually reproduce, especially plants and animals. That is the norm. But there are certain fungi and bacteria that are very efficient at cloning themselves. They don't require a partner to reproduce. So they can asexually reproduce, and that's easier, of course, than having to find a partner and mate. But the two big types of cell reproduction are going to be asexual, that we see in mitosis, and then sexual, that we'll see here in meiosis. When we're talking about sexual reproduction, We have two individuals that mix their genetic information and create something new. This requires the formation of gametes, which is a technical term for the sex cells, egg and sperm. And so we're going to be looking at these two processes in detail. We're going to go into the steps of mitosis and then the steps of meiosis, and definitely be able to compare and contrast the two types of cell division. A little look at chromosomes. Now, I've said this term a few times already, and we looked at chromosomes and this chromosome packaging. It came up a little bit in chapter 4, and this is another topic that we will continue to explore throughout the semester, just this chromosome hierarchy, or how DNA is packaged into a structure called a chromosome. So this image is a really good one to look at and figure out and understand. So eukaryotic chromosomes, remember that's the chromosome in a eukaryotic cell, or cells that have nuclei, is one long DNA molecule wrapped around proteins. We call these proteins histones. So we can see here that the genetic material in our cells is not just naked DNA. It's actually this DNA protein complex. So once that DNA wraps around those histone proteins, that then becomes something called chromatin, that DNA protein complex. Now, when this chromatin then condenses and coils and wraps up tightly into very tightly packaged structures, that then becomes a chromosome. So really the only difference between chromatin and chromosome is how tightly wound up the fiber is, essentially. A really good analogy for this is like a ball of yarn. The entire structure of a ball of yarn would be like the chromosome. But then if you were to unravel that ball of yarn into an individual string of yarn, that unraveled string would be like chromatin. So it's really just depending on how this is packaged, which gives you a chromosome or chromatin state. And it's important to understand that, because in a little bit, when we look at the stages of mitosis and meiosis, as long as you understand what the DNA is doing, you'll be able to figure out what stage of the cell cycle you're in. And all you need in order to do that is to be able to understand, is the DNA in chromatin form? So is it strung out? Is it loose? Is it individual strands? Or has it condensed into thick bands known as chromosomes? So chromatin will condense into its chromosome form right before mitosis and meiosis, or right before that active cell division. And so I'll show you what I'm talking about here in a moment, but that's the importance of this slide, and realizing the two different states that our genetic material exists in and the implications for each of those. So in our cells, humans have 46 chromosomes, 23 from mom and 23 from dad. Different organisms have different chromosome numbers. Like, there's no rhyme or reason to how many chromosomes something has. It just sort of is. Like, a lot of plants have hundreds of chromosomes. I think there's a species of rodent that has like 6 chromosomes total. But there's no rule, okay? But I would know that we humans have 46 chromosomes in every cell except our gametes, or sperm and egg. And we're going to see why there's this exception, and why sperm and egg have a different chromosome count than every other cell in our bodies. So of course, the first couple slides, we established that if one cell has to make 2 cells, everything in that cell has to double, right? So going from 1 to 2, you have to double. So the genetic material also does this. One of the halves of this X-shaped structure here that I'm trying to outline for you there, that is one chromosome. But of course, everything has to double in the cell. So once that chromosome duplicates itself, then we get this characteristic X-shaped structure. So when we see this X-shaped structure, it is correct to say that is a chromosome. But also note that it is just a duplicated chromosome. It's like a photocopy of each half, okay? Because eventually one half of this X is going to go into one new cell, and then the other half is going to go into the other new cell. So just keep that in mind as well as we look at these structures. The center of the chromosome, the duplicated chromosome, will pinch inwards, and this is the site where some little fibers will attach to actually pull these two halves apart. That is called the centromere. And then one half of each X, or one full-fledged chromosome, once it's been duplicated, we can refer to as a chromatid. So 3 new terms to associate with chromosome duplication here. Okay, so now we can jump right in to how cells divide. Just like us, cells have a life cycle. If you even wanted to put the word life right after cell cycle, we're essentially looking at a cell life cycle, if that helps you to visualize this chart any better. It's an ordered series of events during which a cell grows, replicates its DNA, and divides to produce 2 identical daughter cells. There are 2 main components to a cell's life, or 2 main phases, and then those are subdivided into subphases. The majority of the time, a cell will spend its life in a phase called interphase. And as we can see here, 75% plus of that cell's life is spent in this interphase. This is where a cell just does normal cell activities. So the cell is growing, it's just functioning as normal, and then it'll start to think about dividing, so everything in the cell will double, all the DNA will replicate. So the G in G1 just stands for growth phase 1. The S stands for synthesis, or when the DNA copies itself. And then we've got our growth phase 2, when the cell will sort of hit checkpoints to make sure everything is run smoothly, everything looks good, everything is duplicated correctly, and then it will begin to actively divide. The stage of active cell division we call mitosis, or M phase, M for mitosis. And then, of course, mitosis is then subdivided into prophase, metaphase, anaphase, and then telophase/cytokinesis comes at the very end. So make sure you're able to fill out a chart like this in order. Tell me a little bit about what happens in each phase, so like their functions, etc. But for the rest of chapter 8, we are going to be deep diving into M phase, or mitosis. So we'll be able to identify which mitotic phase a cell is in based on what the DNA is doing, and then what overall is happening in the cell in each phase. So here are our stages. So again, we can start with interphase. The chromosomes duplicate. Remember, S phase of interphase is that synthesis of all the material, especially the chromosomes will double now. And it is at this point where we go from interphase into mitosis, or the first stage of mitosis, prophase, where our chromatin, so we're still stringy, diffuse at this point, condenses into full-fledged chromosomes. So we now see, once we have entered mitosis, these characteristics X-shaped structures that we looked at earlier. These are duplicated, condensed chromosomes. So this is a hallmark of being able to identify whether a cell is in active cell division, is look at the DNA, and if you can see thick chromosomes, you know that you're in mitosis, because chromosomes will only be visible when the cell is dividing. If the cell is in interphase, you will not be able to see these X-shaped structures. It'll be in the chromatin state. So once we've entered prophase, our chromatin condenses into chromosomes. And then little organelles called centrioles will start to spit out protein fibers called spindles. Prometaphase is when our nuclear membrane will start to break down, and then those spindles will start to connect to our chromosomes at the centromere, or that center part of that X. Metaphase, so meta which is a Greek term for middle, literally where our chromosomes will line up right on the equatorial plate of the cell. These spindle fibers have now attached to the centromeres of each chromosome, each duplicated chromosome. Anaphase is the shortest phase of all. This is when our spindle fibers retract, pulling one half of each duplicated chromosome to opposite ends of the cell. And then telophase, we can see the nucleus has begun to reform. A cleavage furrow will start to form, right? So that the cell membrane will start to cleave inwards. And then we have our spindle fibers sort of break back down. And then cytokinesis is when when telophase has been completed. So cyto for cell, kinesis for division. This is when we now have two daughter cells from our initial parent. And keep in mind, if we are in mitosis, these daughter cells here at the end should be identical to our parent cell that we had all the way here at the beginning. So here's another really good visual representation of what's going on. Interphase, notice that our DNA protein complex is in chromatin form. It looks really stringy, very diffuse, and at the same time, in an actual cell picture, we can't really tell much of what's going on. Like, we can see the pink nucleus, but we can't really tell anything is happening yet. But then, of course, once we have entered prophase, that chromatin has condensed into duplicated chromosomes, and we can even see here in the actual picture of a cell, our genetic material has started to band together and form thick chromosome structures. So here's another really good picture of an array of cells in various phases. So see if you could go through and determine which phase of the cell cycle every individual cell is in. That would be a good activity for you to do if you wanted to pause this or to come back to it later. So understanding what mitosis is and how it functions is important, especially if you are interested in any sort of cancer biology. So what is cancer? We've all heard of the term, and we probably have all known somebody, maybe even someone in our own family, that has either had cancer or unfortunately has passed away from cancer. It's a very devastating disease, but what is it exactly? Cancer forms when there's a mutation in the DNA that controls that cell cycle. And something in that mutation tells that cell to keep dividing out of control and or does not allow the cell to commit suicide if something is wrong with that cell. So there are a lot of things that have to go wrong all at once for cancer to form. But basically, it originates as a mutation somewhere in the DNA, somewhere in the original programming that controls that cell cycle and those checkpoints that tell the cell everything's good, everything's fine. So, because of that, cells just divide uncontrollably, ignoring important checkpoints. And when this happens, cancer cells can become malignant. So you can have two forms of cancer. You can have benign tumors, which are not harmful. They're just sort of like little growths that can occur. Or you can have a malignant tumor. And that's the really bad one that you don't want. A malignant tumor means that pieces of those mutated cells can actually break off of the tumor and travel elsewhere in the body, which is a real problem because it can spread to your vital organs and shut them down, and then that's what eventually kills people from cancer. And so I've linked a really interesting cancer YouTube video for you if you would like to watch it, just to give you a little bit better understanding of cancer and its biology. So I will put that in our module. Okay, the second type of cell division, meiosis. This is how sex cells are made. Every organism reproduces after its own kind. Sexual reproduction makes unique individuals. So maple trees produce maple trees, but little baby maple trees genetically are different than the parents, the same way that you are not clones of your parents. You are your own individual. We're going to see how that's possible through this process. Offspring inherit a unique combination of genetic information from mom and dad. So we can explore karyotypes at this point. So that's what this image is here to the right. A karyotype is a picture of your chromosome. So this will be a karyotype of a human being. So we have 23 pairs of chromosomes, or 46 total. And every chromosome has two copies, of course, one from mom and one from dad. Chromosomes 1 through 22 are known as the body chromosomes, or somatic chromosomes, or autosomes. And the last pair, pair 23, are known as the sex chromosomes. Pair 23 is the only pair that may not look identical to one another. If you are a human male, you will have one X and one Y. But all the rest, as we can see, they are identical in length, in shape, and size, and also what information is carried on them. So every cell in our body has 46 chromosomes, except for egg and sperm. The matching pair of chromosomes given to us by mom and dad are called homologs. Homo for same, logos proportion or location. So when we see, let's say, chromosome pair number 1, we got one from mom and one from dad. Those are called homologous chromosomes, just meaning it's chromosome number 1 from mom and then chromosome number 1 from dad. So again, just to reiterate, if we were looking at a karyotype and breaking down the information, this is all that means. We have 23 pairs of chromosomes, one from mom, one from dad. Those are called homologs or homologous chromosomes. So 23 pairs of homologs. And then chromosome pair number 1 through 22 are autosomes or body chromosomes. And then the last pair, pair 23, are the sex chromosomes. Because we have two copies of each chromosome, one from mom, one from dad, in every cell of our body except egg and sperm, we have to be able to differentiate between how many chromosome copies are in a cell, for example. And there are only two possibilities. So if both copies of the homologous chromosomes are in a cell, we call that a diploid cell. Di for two, ploidy for chromosome count. We can also abbreviate diploid with shorthand, a 2N. 2, of course, for the two copies of each chromosome. But if only one type of chromosome is present, or there's only one copy, we call that cell haploid, or half. There's only half of the genetic information. Now, there are only two cells in the human body that are haploid, and those are the egg and the sperm, or the sex cells. Every other cell, literally, is always going to be diploid, meaning it's got a copy from mom and dad. How we go from diploid to haploid and back again is called a life cycle. This is the human life cycle. So if we start at the top, and of course you can follow the color coding here if you are confused. Egg and sperm are gametes. And all that means is that they are sex cells. Sex cells are haploid, or they only have 23 chromosomes each. There's only half of the genetic information in these. So that's why we call those haploid cells. Egg and sperm will fuse during a process called fertilization. And then what happens if you add 1 plus 1, or 23 plus 23? Then you get 2, or 46. So we see that once egg and sperm fuse in this process of fertilization, we have changed the ploidy level from haploid to diploid. And then we begin a new human life cycle. So we have a diploid zygote, that's our one-celled self, undergoes mitosis and development. Then we become adults and reproduce on our own. And then in specialized organs, whether you're male or female, cells will undergo the process of meiosis to make sperm and egg again, and then on the life cycle continues. So you can see that life cycles are nothing more than alternating between haploid stages and diploid stages. And then all that means is how many copies of each chromosome is in the cell? Just 1 or 2? And the only time you ever have haploid cells is in the gametes, the egg or sperm. Everything else is always going to be diploid. But meiosis, that we're getting ready to explore, is literally when we go from a diploid cell to that haploid cell. And that is a special process because there's a lot that has to happen. And there's also a lot that goes into making every single egg cell and every single sperm cell a little bit different than the other. And there are trillions and trillions of sperm cells that a male makes throughout his lifetime. And there are hundreds of thousands of egg cells that a female makes throughout her lifetime. And every single one of those is a little bit different through this amazing process called meiosis. So I would be able to fill this life cycle diagram out and understand what's going on here, how this is alternating the ploidy level from haploid to diploid and then back again, and what that means. So let's go through meiosis now. And the good thing about this is that if you understood mitosis earlier, you're going to understand meiosis. There's just a few extra things that happen in meiosis and not mitosis, but it looks pretty similar. There's not too much different, just a few extra little steps. So that's always good news. We have to produce haploid gametes to keep our chromosome number from doubling every generation. It's that process of cutting the genetic information into half that is meiosis. So the first thing that happens, of course, same thing in mitosis, is that our chromosomes duplicate during interphase. And this is something new that didn't happen in mitosis. Homologous chromosomes will line up together. So mom's chromosome number 1 will line up with dad's chromosome number 1, all the way down through the 23rd pair. So our homologs find each other. They don't do this in mitosis. They just line up however. They don't need to find each other there. But here they will. So we have this double X feature. Instead of just one X, we now have a pair of duplicated homologous chromosomes. So the first thing that happens, okay, once our genetic material has duplicated and our homologs have lined up, the homologous chromosomes separate, meaning mom's material goes into one new cell and dad's material goes into another. It is at this point, during meiosis 1, that we have officially gone from diploid to haploid, because now we only have one copy of each chromosome per cell. And then meiosis 2 is just basically you separate your chromatids. So this is pretty much the same thing as mitosis at this point. So we can see that meiosis is a double round of cell division. First you separate the homologs, and then you separate the chromatids. So that at the end here, you have 4 haploid gamete cells. And whether you're female or male, these would be egg cells or sperm cells. And as you can see, each egg cell may have only mom's material or dad's material. So we have literally cut the amount of genetic material in half by the end of this process. So here's another representation of that. We have our chromosomes duplicating, and then we have our homologs lining up. Meiosis 1, our homologous chromosomes will separate. So here's mom's chromosome 1, here's dad's chromosome 1. We're in separate cells now. This will be, let's say, mom's chromosome 2, dad's chromosome 2. This would happen 23 times over. This is just simplifying that for aesthetic purposes. And then meiosis 2, we just separate our chromatids. But that big ploidy level change happens right here in meiosis 1, when mom and dad's homologs split apart. And there may be something else interesting that you notice about this image. It may be that after meiosis 1, we notice that our homologs look a little bit differently than they did before. Or it looks like pieces of each homologous chromosome has broken off and then reattached to the other. And that's indeed exactly what has happened here. Because as we can see, our resulting egg and sperm cell has different genetic information than it did in the beginning. So this is one really big way that every egg and sperm is a little bit different. The homologous chromosomes have hot spots along them at certain points that can break and then reattach to one another, literally mixing and mashing the genetic material to give you something new from something old. And that really is the basis of sex, of sexual reproduction. It's quite beautiful if you think about it. But that is what's going on here with the different colors in the chromosomes at the end versus before. And there's a name for this, and we'll come to that in a moment. Oh, let's skip the slide here. That slide, it's not letting me go back, but that was just a side-by-side of mitosis and meiosis. But then let's look at the process I was describing a moment ago, a little bit more detail. So our first big difference between the two types of cell division is that meiosis cuts the chromosome number in half using that double rounded division. And then, of course, meiosis then creates new genetic material, new combinations, I should say, from the old. And it does this in two ways, crossing over and independent assortment. And what we saw earlier with the pieces breaking off and reattaching, that's called crossing over or genetic recombination. So again, you have to imagine there's 23 pairs of these in an actual cell. We're just simplifying by showing one pair. Along certain points, all right, are hot spots where a piece of a chromosome will break off and then reattach to its homologue, so that eventually you get new combinations of chromosomes that you didn't have before. So you have new versions of parental chromosomes created. Another way meiosis creates genetic diversity is how they line up on the equatorial plate during metaphase. So they independently assort from one another, so that eventually you could have egg cells with only mom's information, only dad's information, or a combination of the two. It just depends on how they line up. Are all of dad's on the right and all of mom's on the left? Are they mixed and mashed, etc.? So understanding meiosis a little bit better can help us to understand what happens when it goes wrong. So just how cancer is a result of mitosis going wrong, genetic disease is what happens when meiosis goes wrong. So something has happened to that cellular machinery, either in meiosis 1 or 2, that does not separate the genetic material properly. So genetic disease, that's essentially what has happened here. The egg and the sperm have an not normal number of chromosomes, and we call that aneuploid. So the failure to separate either homologous chromosomes in meiosis 1 or sister chromatids in meiosis 2 is called non-disjunction, literally not being able to separate apart. So we have two scenarios because, again, we have 2 rounds of meiosis. So if this were to happen early on in meiosis 1, when our homologs are supposed to separate, so we can see here something has happened. That cellular machinery did not do its job and did not separate this pair of homologs. So then the resulting cells, all right, look something like this. And then we can see after meiosis 2, every single egg or sperm down here at the bottom is abnormal. So some of them have an extra chromosome, so that's what that +1 is. Some of them are missing a chromosome, that's what that -1 is. So if non-disjunction happens during meiosis 1, nothing is going to be normal. That's the worst case scenario. But if it happens during meiosis 2, look what happens here. We have one cell with an extra, one cell missing, but you've still got a 50-50 chance of having normal gametes if it happens later on. So if you had to pick between the two, obviously non-disjunction in meiosis 2 is the lesser of 2 evils there. But this just results from something in that cellular machinery not working correctly or breaking down. Something has happened to give us that genetic disease or an abnormal number of chromosomes. So we call that aneuploidy, abnormal ploidy level. So here we have a normal karyotype. So a diploid human has 46 chromosomes. Everything's good. But then we have some examples of aneuploidy or genetic disease. So we can have a human missing a chromosome, so 2n - 1 or 45 total, or a human with an extra one, right? That's what that plus one is. So 47 total instead of 46. Normally, okay, it is better to have an extra chromosome than to be missing one. Oftentimes, if you are missing a chromosome, that baby, that zygote, or even fetus, if it makes it to that point, is not viable. It cannot live long missing a chromosome. But having an extra chromosome is not necessarily a death sentence. So, again, if you had to pick one of the two, it's more viable to have an extra chromosome than to be missing one. Especially aneuploidies in chromosomes 13, 15, 18, 21, and 22. Those people can live with extra chromosome numbers of those, and definitely the sex chromosomes, because if you have aneuploidy in the sex chromosomes, you may not be fertile, you may have different physical characteristics, but it usually is never a death sentence. So, for example, all right, one of the more common genetic diseases is called Down's syndrome. I'm sure we've all heard of Down's syndrome. And what does that mean? Well, Down is just the name of the doctor that really coined this abnormality. But the scientific term for Down's syndrome is trisomy 21. And what does that mean? It means the somatic chromosome, so the autosome number 21, has 3 copies instead of 2. So tri for 3, somy for the body chromosome, and then body chromosome pair number 21. So Down syndrome people have an extra 21st chromosome. So they would have 47 total instead of 46. It's quite common, actually. 1 in every 800 births, and it actually is a spectrum of outcomes. So it's a range of abilities and physical characteristics. Some of them are more severe, some of them are more benign, but overall, individuals with Down syndrome are able to go on and lead very normal and fulfilling lives. And then some examples of sex chromosome aneuploidies. Of course, here we have a normal human female with the sex chromosome complement XX, 1 X from mom, 1 X from dad. But if we have aneuploidy, let's say of 45, so they are missing that second X chromosome, that N-1 there we saw. Only 1 copy of the X chromosome. That is called Turner syndrome. And these individuals are sterile, meaning they cannot go on to make egg cells and reproduce themselves. What about something like an extra chromosome? So here we have the N+1 aneuploidy. So something like Klinefelter syndrome. We have a human male that has an extra X chromosome, so XXY instead of XY. These individuals are also sterile. So again, the sex chromosomes, as we can see, really have a lot of influence in reproduction and health. And if you are aneuploid in the sex chromosomes, it's not a death sentence by any means, but it's going to greatly affect your ability to go on and reproduce. And that's all I have for Chapter 8. Again, as always, make sure you read the actual chapter in your textbook a few times. Watch some YouTube videos, and as always, please reach out to me if you have further questions. Thank you.